Greetings. The podcast that you're about to listen to is a phenomenal example of a group of students who have synthesized an advanced sort of piece showing their connections between what we have studied throughout a semester of biochemistry and the problem of water contamination. Absolutely one of the greatest problems facing our world. So please keep tuned in as this will convert to being audio only. And I hope you really enjoy this spectacular example of student synthesis.
Hello everyone and welcome back to the Hydro Safety Network, your spot for water thought. As normal, I'm your host, Michael Downstream, and today we have a fantastic episode today. Joining me today are Bailey McFlorate.
Hi everyone.
And Caleb Hydrophobe.
Hey.
Guys, would you like to discuss your backgrounds a little bit for us?
Sure, I have a background in chemistry that I studied as an undergraduate, and now I look a lot into proteins.
And my background is in chemistry and biology.
And then for those of you who don't know about me, I have a background in chemical engineering and chemistry. And today we'd really like to discuss the underlooming water pollution problem. And this is important because water is so essential for us in our day-to-day life. And we'd really like to give you a little bit of a brief background of water and why it's so important for us. And it's important in a variety of ways in our lives. It's an essential part, and it is the thing of hydrolysis. And it's also a key component in protein folding. And Bailey, just because you have a little bit more of the biology background, would you like to discuss hydrolysis for us and why it's important in the body?
Of course. So for members of the audience that does not know, or that do not know, one of the key things about water is that it's a universal solvent, which simply means that it dissolves more things than most other liquids. But there are still things that it does not dissolve. So, of course, going into hydrolysis, if we break down the words, we hear hydro, which it means water, and lysis, which means to break or separate. And it is a key component of the different reactions that occur within the body. Enzymes use hydrolysis to produce molecules for our everyday energy, and within this we use it for citric acid cycle, and in the citric acid cycle, we end up adding one OH and hydrogen to each molecule through nucleophilic attack of lone pairs on electrons to an electron-deficient carbon. This will then break off the OH group, leaving a positive hydrogen behind, and a negatively charged location elsewhere on the product. And as we well know, the opposites attract. So the hydrogen bond will bond to a negatively charged molecule. And this allows for crucial molecules such as like NAD+ and NADH to undergo oxidation and reduction reactions that will be used to power future citric acid cycles and can continue to provide us with the essential energy we require every day.
So Bailey, you mentioned a couple really cool parts of biology and chemistry there. You talked a little bit about the ability of water to dissolve things and a little bit about enzymes and NAD and NADH, which are actually called cofactors.
So, Caleb, what is a cofactor or a co-substrate? Why are they important in enzymes?
That's a wonderful question. So cofactors are molecules that help enzymes function. They bind to them and through complicated chemical pathways cause reactions to occur, such as hydrolysis. And enzymes themselves are proteins. So to understand proteins, we have to look at what they're made of. And proteins are composed of amino acids, which are little molecules. There's about 20 of them that we need to live, and they come in all shapes and sizes. And most of these are either polar or nonpolar, and that has to do with water being able to dissolve things. So nonpolar means that water will not dissolve it very well, and nonpolar molecules tend to prefer hydrophobic areas. They're hydrophobic, which means that they are afraid of water. The more polar amino acids tend to go into water more easily. So what these amino acids do is they form long chains and link together, kind of like Legos, you could say. So like, I'm sure many people have played with Legos. You put them together to make something cool. So if you were going to make a Death Star, which is composed of lots of Legos, it'd be kind of like making an enzyme, which is made of a lot of amino acids. And what's really cool is that because of water, these amino acids will fold on each other in the exact same way. So it's like those Lego pieces being formed into a Death Star the same way every time. It's really cool how it works. And that's because of those hydrophobic regions, which will tend to go inside the enzyme, and the hydrophilic regions, which will tend to be on the outside of the enzyme or the protein. It's really cool how water affects even the most basic fundamental parts of our bodies.
Okay, guys, thanks for that background. And back to kind of the core issue of today, we'd really like to talk about water and why it's so important to discuss this water pollution problem. Recently, I was reading something in the New York Times that was discussing why water is actually becoming a very scarce resource for us. We're seeing that roughly 25% of the population is actually becoming deficient in water. We're actually having too many droughts and too many other issues where we're depleting our water resources very, very quickly, quicker than they can actually be replenished. And with this growing need, what we've seen from an engineering perspective is actually a turn towards wastewater reclamation. So I'd really like to discuss this idea of wastewater a little bit more and how we can improve this idea to help ourselves out with this water pollution problem, and just the general idea of some areas where it actually kind of fails us. Wastewater is essentially a water supply from a community after it has been used in a variety of ways and applications, and it's really just kind of dirty. It contains a lot of constituents or particles and other matter that kind of render it unsuitable for consumption or use through other processes now. This is why we need to treat the water. It's not basically unusable. And these waters are usually rich in organic compounds and in nitrogen and phosphorus containing compounds. And this is vital for life, but in too high of a concentration, they're actually very detrimental to us. And so a typical wastewater treatment plant will use its own microenvironment, and it actually uses an anaerobic and aerobic area, so areas without oxygen, anaerobic, and then areas with oxygen. And it uses a bunch of little bacteria in both of these areas to kind of eat the organic matter and use these nitrogen and phosphorus compounds to grow and live. However, they don't do enough on their own. We also have to use some physical and some chemical properties to help us separate out the constituents that we have here. And these are ideas such as filtration, some separation, adsorption, and then just the general disinfection. And as our awareness of these harmful constituents continues to grow, the technology for treating this wastewater actually has begun to lag behind, and it's not been able to keep up with the increasing number of issues that have been arising over the last 30 years or so. And one of these core issues that wastewater fails in is in this microbiome. It's not actually able to remove all of the organic constituents from the wastewater. We see that even our capabilities of detecting some of these dangerous constituents are to a point where we can detect 1 or 2 parts in every trillion, or like 1 gram in every 265 million gallons of water, to put that into perspective for you. And what this problem does is it leads to something called eutrophication. And eutrophication is really just an overgrowth of these natural animals and these natural organisms that exist downstream of a water treatment plant. And these blooms cause really 2 major issues. One, we see an overgrowth of life, and what this does is actually cause a depletion of oxygen in the water, and oxygen is almost essential for all life. And this overgrowth of plant life actually leads to a mass die-out in this area because the water will no longer have oxygen in it. And recently we've also been able to develop further understanding of other issues that have occurred due to this eutrophication problem. And Bailey, I understand that you've actually been researching this topic recently and have some interesting findings. Could you share with us some of what you've discovered?
Of course, Mike. So one of the biggest thing is when we have pollution going into the waters, we have other different like nitrogen, phosphorus, and it's leading for a lot of different growths, including in like algae, dinoflagellates, and like phytoplankton.
So what are like dinoflagellates and phytoplankton for those of us who really don't know?
They're little tiny protists that live in water. So they're little tiny organisms. And if you want to think of plankton off of SpongeBob, the little green guy, that's a good description of what they are. They're little tiny creatures that can cause a world of problems. And within these like algal blooms, which are just the overgrowth of these plankton, we have some that are creating toxins. And one of the biggest toxins they are creating is okadaic acid, or OA. Okadaic acid is responsible for diuretic shellfish poisoning, or DSP. So DSP causes vomiting, diarrhea, and nausea, which is already a lot of fun for anyone who's ever had food poisoning.
No kidding.
It's terrible. But it turns out they can do even worse things to different animals and humans. So an article published in Marine Drugs called Okadaic Acid, More Than a Diuretic Toxin, goes into these different things it can do. Okadaic acid acts as an inhibitor for many serine and threonine active sites on proteins. So basically, when these enzymes are trying to function, it will go in and attach to serine and threonine and prevent them from acting the way they're supposed to.
And serine and threonine are just some of those amino acids that we talked about, right?
Yes, they are. So some of these proteins are phosphatases, which the main one we're going to be discussing is PP2A, but we will also talk about PP1 and other types. But okadaic acid has the strongest affinity for PP2A, which the highest affinity just means that it really loves PP2A.
So what is PP2A? Like, what is this enzyme doing in our body?
Of course, let's go into some backgrounds because many of you might not know. So PP2A simply means protein phosphatase 2A. So it's just a type of phosphatase. And it is involved with tumor suppressing functions, which is really important, especially if we're going into and looking at cancer. So in the journal Cancer Letters, there's an article that was published that describes the importance of PP2A. So it is a serine threonine phosphatase, so it goes and attaches to serine and threonine, and basically it takes off the phosphate groups from them. So how this is done is basically PP2A goes and does a nucleophilic attack on phosphoserine and phosphothreonine. So those are just the regular amino acids with a phosphate group attached. And these attacks can only be done in the presence of water. And it actually plays a vital role in the cell cycle, programmed cell death, glycolysis, cell metabolism, DNA replication, transcription, and translation, and so much more. So it's a very important enzyme.
Could you discuss with us some of these terms that you just threw out there? You discussed a lot of different things. So what is something like transcription or translation for some of us who don't recall our freshman biology?
Of course. So with transcription and translation, they're within the central dogma of gene expression. So when we go through and replicate our DNA, it acts as a template so we can make mRNA, so a type of RNA. And this is then translated into amino acids and proteins, which give us different traits and benefits. So along with that, it plays a huge role in tumor suppression. As you may recall, I said it plays a role in programmed cell death as well as a cell cycle. So if it's inhibited, we can have an overgrowth of cells, which produces tumors. And these tumors tend to form in the lung, breast, skin, and colon. So those are the types of cancer it also causes. So with PP2A, it has two different forms, a dimeric and a trimeric form. And the trimeric form means it has 3 different subunits, which are just chains of amino acids that are non-covalently bond to another chain of amino acids. And this trimeric form is actually an active holoenzyme complex, which a holoenzyme is just an enzyme that also needs a coenzyme to work. So it needs an additional helper to come in so it can be activated and end up working. So okadaic acid inhibits the activity of PP2AC, which is a specific subunit off of that. So what that basically is doing is it's filling that slot so PP2A cannot take off phosphate groups. And while okadaic acid is not classified as a known carcinogen, in different animal experiments it has been shown as a tumor promoter. More studies are being done to determine if humans that are repeatedly exposed will develop cancer.
So what are some of these animals that show cancer? And Caleb, I think you might have had something to say there. What'd you discuss?
I was listening to what Bailey was talking about, and she mentioned how some enzymes have, or this okadaic acid has a higher affinity for PP2A than maybe some other enzymes. And if you can imagine how enzymes work, they have something called kinetics. And so if you can imagine a bunch of them floating around in a solution, they have their substrates that float around as well.
So what is the substrate? I mean,
So in this case, it would be okadaic acid, which is not the intended substrate, right? But the intended substrates will be there as well too. Otherwise, the enzyme couldn't really catalyze anything. And so when you say that there's a higher affinity, what you're really saying is that an enzyme has a lower Km value. So Km value pretty much just says that there doesn't need to be as much substrate in there for the enzyme to bind to it. And enzymes are pretty much defined by this Km value, and it's a lot more complex than that, but there's also the maximum velocity or the rate at which an enzyme can catalyze a reaction. And so a very effective enzyme that is catalyzing a lot of reactions would have a high maximum velocity and a low Km, which would mean that it has a great affinity for its substrates and catalyzes them very quickly.
one other thing I think we should mention about this is that enzymes are actually very particular about what is a substrate and what they can actually do. So most enzymes are very actually specific. So think about like a screwdriver. We can't just use a Phillips head screwdriver for every single screw and nut and bolt out there. We have to use the right tool for the right job. So when we talk about substrates, it's usually some component or some other thing that is able to specifically fit in this region of the enzyme. And it may not be the intended thing that's supposed to fit in there, but it is going to affect very specific enzymes because of the way it is shaped.
And that's really what okadaic acid does. It has a perfect shape for PP2A, so it can easily fit in there and block its actual substrate from getting in.
Perfect. Awesome. So let's bring this back to kind of what Bailey was talking about a little bit. What are some of the animals that we've actually seen this effect in, this idea that it promotes tumor growth in?
So of course we've been using mice and rats. They're kind of the go-to on any experiment, especially when you get into animal trials. And what it has shown is that when we're using mice skin, they're developing a large amount of tumors. And when we look at rats' stomachs with okadaic acid, they're developing a lot of different tumors in their stomachs as well. So it's just showing an overall promotion of tumors within the skin, the stomach, and different areas like that.
So do we really know how humans get exposed to this? How is this a problem for humans?
Oh yes, we do. So okadaic acid is accumulated within like shellfish, bivalve mollusks, and fish. So stuff we commonly eat. So they eat it, the different plankton that have and produce okadaic acid, and then it gathers in their bodies. And then, of course, we then eat the fish, the shellfish, and like oysters, mussels, and stuff like that, which is how we end up getting sick.
So then, is there any way that we are able to denature this toxin? I understand that with a lot of proteins, for those of us who don't necessarily understand denaturization, it's an idea of we can either change a temperature or a chemical property of this system, and it'll actually make this toxin basically inert. It makes it so it won't react in our bodies. Is there a way that we can cook the shellfish or something like that to prevent this from happening?
So unfortunately, no. That's actually why okadaic acid is so hard to deal with. Because no matter how much you cook the fish or anything else that you're eating that has been contaminated with it, it won't denature it. And even if you freeze your food before eating it, it will not have an effect. You can still get sick from it. And the interesting part about this is that we're not exactly sure how okadaic acid causes diarrhea. So the Marine Drug article actually theorized two different ways. The first is that okadaic acid induces the contraction of smooth muscles by basically, again with phosphorus, it's preventing it from being dephosphorylation. So basically we have a bunch of phosphorus that's causing all the muscles to contract within the small intestine and the gut, which is causing diarrhea. And the other way is it's having a lot of phosphate on proteins that regulate sodium secretion. So it's basically preventing them from doing their job. And your body's looking at all these different levels and it's like, geesh, we need to do something within the small intestine to fix this problem, which then this body solution is to cause diarrhea. And within like infected humans and animals, we still have amounts of OA even after the food has been through our digestive system. So it's not even denatured within our guts. And another alarming issue that's actually happening in Europe is they've set a regulation for the fish and shellfish on the market where they have to have at very maximum 160 micrograms of OA equivalent per kilogram for fish and shellfish, which is actually a lot considering only 40 micrograms will cause vomiting, diarrhea, and other unwanted symptoms. So basically, by allowing this fish and shellfish to continually be on the market with amounts, we're introducing more and more into the human body, which puts humans at risk of chronic exposure, which may actually lead to cancer in the end.
Wow, that's
Sounds like water. The pollution of water is causing a lot of problems these days.
Yes, it is. It is literally causing worldwide amounts of toxins to increase and people to become ill.
wow. Thank you for that insight and understanding of why part of our problem with this water pollution idea and how can we help control wastewater and how we can continue to improve upon these ideas that have not been perfected yet. And I'd kind of like to continue our discussion about wastewater and some of the things we do to treat it. Earlier I mentioned that it's not only the marine biome in wastewater, but we also used to do something called chlorination. And this is really the idea of we would add some chlorine-containing chemical to disinfect our water and sterilize it.
So is this kind of what they do to pools then?
It's a similar idea, but not the exact same.
Okay.
But it's effectively the same idea.
So how is this different from, say, a co-substrate or something working with an enzyme?
So this is a little bit different because this is designed to actually just kill the microbes. It's designed to basically be a toxin, some form of a poison, so we can get rid of other organisms that might actually cause problems in humans. That was the key idea of this. However, this had other unintended consequences. For several reasons, we did want to add this chlorine. There are a lot of microorganisms crawling around in wastewater, particularly in domestic wastewater, so the stuff that you're flushing down your toilet. A lot of that comes from humans, and there's a lot of human disease that can very easily be spread. So there's a lot of incentive to get rid of a lot of these naturally occurring bacteria. The problem is that chlorination has unintended side effects. And these actually result in reactions and syntheses of unintended chemicals in our water. And some of these compounds are known as trichloromethanes, haloacetic acids, and several other classifications. But the most pertinent one that we've actually discovered so far is a chemical known as N-nitrosodimethylamine, or NDMA. And this has been found to be the product of chlorination in water, and is actually very dangerous due to its mutagenic properties in the human body. And for those of you who don't know, it just means that it's able to affect our DNA in negative ways. And it does this because it actually undergoes something called hydroxylation. And for just revisiting our background, that's actually a part of hydrolysis. This hydroxyl group is a negatively charged OH that comes from water. And this hydroxylation makes a very unstable intermediate compound, and in the liver this actually decomposes into formaldehyde and monomethylnitrosamine, which is a very dangerous chemical because it's highly unstable and mutates into an even more unstable molecule known as methyl diazonium ion. And this ion has the ability to do what's known as methylation, or the alkylation of DNA. And the reason why this is a problem goes back to the chemical structure of DNA. For those of you who don't remember, DNA is the effective genome of your body. It's what tells your body who you are and what to do. And it's comprised of what's known as an alpha helix. So it's going to make a big spiral out of two chains that wrap around each other. And the way these two chains bind together is known as something called hydrogen bonding. And what this methylation does is it actually prevents the hydrogen bonding in between two of the molecules in DNA. So DNA is made up of 4 key components. adenosine, guanine, cytosine, and thymine. And these can be abbreviated as AGCT. And so but the A and the T love to bond together because they actually form 2 hydrogen bonds, and the G and the C like to bond together because they are able to form 3 hydrogen bonds between one another. And so this methylation actually occurs on the guanine. And what this does is it actually prevents one of these hydrogen bonds from occurring. It can actually allow DNA to be misrepresented because we can only have 2 hydrogen bonds active. And so when this happens, it actually allows for a substitution of one of these base pairs into one another. And so this actually causes cancer if done in the wrong spot because our DNA does not know when to stop. It just continuously goes on and on. Yes.
So
Caleb?
you mentioned that it causes cancer, and that's an easy word to say these days, that this or that causes cancer. Everything causes cancer. But this is actually one of the principal causes for cancer is when DNA is no longer able, through that central dogma that we talked about, to form the proteins that it codes for. And a lot of these proteins that when we say it causes cancer, it means that the proteins or the enzymes that prevent cancer from happening are no longer being formed because those genes are not coding for them anymore. So you can imagine how or why the methylation or what you were talking about, Michael, could be extremely bad scenario for life in general.
Absolutely. And actually, this is one of the most common products that we see in cancer patients as these methylated groups. It's actually one of the most common unexpected things that we see in the urine and other things in cancer patients. And so one of the things with this wastewater engineering problem has actually been the transfer from chlorination into UV radiation to treat our water. And this is a major improvement because it does several things for these plants. The UV lights are actually cheaper to run than adding chemicals most of the time. A second thing that it's done is that when UV is done properly, it is just as effective, if not more effective, at killing the microorganisms in the water that we do not want to be exposed to. Third, without this need for potentially dangerous chemicals, we don't get exposed to a lot of these dangerous reactions that are going to occur in the body. And lastly, UV radiation has actually been found to destroy some of these harmful chemicals that are actually produced through chlorination. It has actually been found that UV radiation actually is able to form these unstable reactants in NDMA and actually remove NDMA from water as a whole. And this is actually seen when it's vaporized too, because when it's exposed to sunlight, it actually only has a half-life of 5 to 30 minutes in air. And this is a fairly new topic, and yet I understand that there have been dangerous chemicals and dangerous additives that have been going on in water for quite a long time that we really have been arguing about for an extended period of time and multiple decades. And Caleb, I understand that you've been doing some research about this. Would you like to talk about what you've been finding recently?
I would love to, Michael. So until now, we've been talking a lot about how wastewater is treated and how that can cause overpopulation and blooms and stuff like that then produce dangerous chemicals and stuff like that. But a lot of the chemicals that get into water as well that can cause a lot of problem come from fertilizers and pesticides and herbicides, stuff that we put into nature to make a favorable advantage for us that then get washed into water, into a stream or something like that, and then cause a host of very bad problems. So one example of this is the use of organophosphates. So organophosphates are useful in fertilizers because they do really good at what they're intended to do. However, that's bad because when the excess organophosphates get washed away, they keep on doing what they're intended to do.
So is that just stimulating plant life and the growth of life? Like, what is the significance?
Quite the opposite. So a lot of organophosphates, as we've discussed before, are inhibitors of enzymes. So there can be a couple types of inhibitors. Enzymes have things called active sites and also allosteric sites. Allosteric sites can be where some of those cofactors or coenzymes, allosteric cofactors bind to an enzyme to make it work better. The active site is where the substrate actually binds to the enzyme and is converted into the products.
So can inhibitors affect both of these sites?
Yeah,
or
of course they can. So Some inhibitors are called competitive inhibitors, which are at the active site of the enzyme, and some are called non-competitive. And there's a couple other different types of inhibitors, but generally they work differently with the kinetics of enzymes. Would you like to say something, Bailey?
Yes, of course. So when you're saying non-competitive, are you meaning like where do they attach at on the enzyme?
So competitive or non-competitive is basically saying that it is not competing with the substrate of the enzyme at the active site. So it is preventing the enzyme from working in some other way.
Attaching to a different area on the
Yeah.
enzyme.
And that just changes the shape of the enzyme and
Makes
prevents this active site from being open, basically.
Exactly. A perfect example would be hemoglobin in your blood. There are a couple of allosteric inhibitors that attach to hemoglobin that make it less likely to bind to oxygen. And those are intended, so that can actually be beneficial to your body. But what competitive enzymes do, or competitive inhibitors do, is they make it look like the enzyme has less affinity for a substrate, so that Km value will increase. And there's now more substrate is needed in order for the reactivity to continue. While non-competitive inhibitors make the actual overall reactivity, catalytic ability of that enzyme to decrease. And some types of inhibitors can even make the enzyme look like it has more affinity for a substrate, but inhibit the enzyme altogether anyways. Yeah, but continuing on the discussion of fertilizers and organophosphates. Organophosphates can act as inhibitors to enzymes. And one example that we see these days is called chlorpyrifos. This can disrupt the nervous system of marine animals because they just don't have an enzyme like a lot of mammals do that live on Earth that decomposes organophosphates. However, one of the things that I've been looking at more often and discussing in literature and in my scientific study is the use of glyphosate. So glyphosate is an organophosphate that is used in something called Roundup and a lot of other herbicides.
So this is a huge issue right now.
Yeah, Roundup is a very common, I'm sure many of you have heard of it, it's a very common herbicide that you just spray onto plants and it kills it. And it's very effective because what glyphosate does is it disrupts an enzyme called, it's got a really big name, so I'm just going to give you guys the short acronym, I guess you could say, for it. It's called EPSPS. So what EPSPS is it's an enzyme that catalyzes phosphoenolpyruvic acid. And yeah, that's a lot to take in right there. Pretty much it leads to the formation of aromatic amino acids in plants, fungus, and bacteria, but not in animals. That process does not happen in animals. And so what happens is glyphosate looks a lot like phosphoenolpyruvic acid. In fact, it is actually just glycine, which is an amino acid with one slight modification on it, where instead of having a hydrogen on the amino group, it actually has a phosphomethyl group, which, as you can see, could be very deceptive. And since it looks so similar to the substrate of that enzyme, it just binds to that enzyme and permanently deactivates it. So it's not like it decreases its affinity like we've been talking before or inhibits it in that way. It just deactivates the enzyme and doesn't allow it to work anymore. And since it's not working anymore, the plants cannot create these amino acids that they need, and eventually they just starve to death and die. And why it's so popular is that it supposedly has a very little effect on animals, since this process doesn't happen in animals. But as we've been seeing recently, and you may have heard in the news, a lot of cases have come up how it is actually affecting people and animals, and it can cause cancer.
So, Caleb, I've looked into this a little bit just because I am a chemical engineer. And and one of these major companies is actually being sued. Is it necessarily the glyphosate that's causing this issue, or is it something else that they're doing to this?
That was a wonderful question. So a lot of people have been studying this for years. It's been around for about 4 decades. It's one of the most common organophosphates used these days. So a lot of people have looked into it, and if it was causing cancer, we really want to know. So one particular study that I've been looking at is hold on, just let me
Find it real quick.
find it real quick so I can give you guys the name of the author. His name is Yu Hao, and they live in China. They work for a.
Okay.
Did I say that right?
Maybe.
I don't know. But this guy does some very good work on glyphosate, and they published an article in the Journal of Environmental Science and Health where they talk about how they see what the effect that glyphosate has on some of the human strands of like lung cells, liver cells, and other types of cells in our body. And what they found is that glyphosate itself doesn't necessarily cause cancer or kill these cells, but some of the other things that they put in the Roundup and some of these, or specifically Roundup is what they did in this study, actually kills human cells. So
I understand that, you know, a lot of the time, a lot of companies will use their money to promote something that says their product is fine. Have you found issues, especially with a major legal case like this, finding really good science about this, or have you found a lot of biases in this?
You know, that's a very good point, because a lot of like, there's an overwhelming amount of literature that has been conducted over the last few decades that points towards the fact that glyphosate is completely harmless and that it's actually one of the best ones to use because it doesn't affect people. But you go and look at these sources and stuff like that, and you find that the people paying them to do this research are the people that are making money off of glyphosate. So how can you publish something that is going to hurt the people that are paying you? It's kind of like biting the hand that feeds you, right?
Yeah, and I think that's something that we often kind of overlook with scientific literature. We don't really dive into a deeper understanding. We just look for something that says what we want. We have our own biases. I think that's something that we often struggle with. And I think that's kind of part of the underlooming issue with this water pollution problem is, for example, I recently was able to go to New Zealand, and it's amazing to see how in a lot of these more remote areas that are away from humans, just how crystal clear this water is and just how blue and just natural looking it is. Whereas when you drive over something like the Mississippi River, it's all brown and murky, and you don't really realize just how much pollution we've added to water until you actually see these stark differences. And maybe that's something we should dive into a little bit more here just for a few minutes. Bailey, do you want to talk about your sources that you've used at all?
Yeah, of course. So on my one source from the marine drugs, the school that it was done at was the University of A Coruña. I don't know actually how to say that,
Okay.
but we're going with it. Anyways, it's in Spain, and they're actually funded by the Spanish Ministry of Economy and Competitivity, which is kind of a weird source for scientific research at all. And I think what's really going into this that might end up being a bias is that we're talking about regulations within shellfish and fish and how this toxin affects it. So it might have an economic, basically, disadvantage to publish a bunch of stuff saying that the fish is toxic because less people would actually buy it. But again, this isn't really discussed by the source or anything, so that's just kind of a guess of what might be a bias within it.
Okay.
And then the second one that was about PP2A, it's actually funded by the Department of Defense and the National Institutes of Health, which this one makes a little more sense because we are talking about a tumor suppressing enzyme and the possibility of decreased cancer. The Department of Defense doesn't make as much sense, but both of these, since this article is a very generalized discussion on how things are functioning, I don't foresee there being that much bias in it.
One really refreshing thing about this paper that I was talking a little bit about with you guys is they even state at the bottom or at the end of their paper that there is no conflict of interest in the writing of that paper. And it's really refreshing to see how some science is done correctly, and when it is done correctly, the results are reliable. Whereas you can go look. I know I was looking the other day. I was researching glyphosate, and there's something called a glyphosate task force in Europe, and they had this part on their website that cited. They said over 1,000 literature articles that say that glyphosate is completely harmless. But when you go look at who they are and what they stand for, of course they're going to be citing literature that says that.
Because they're actually producing what contains the glyphosate.
Exactly.
They're benefiting from it.
It directly benefits them. Whereas this paper that I'm citing is from a school of pharmacy in East China. And they directly state, and they even cite papers that also state that they have no conflict of interest. And that they are finding that detrimental effects can happen to humans and to other animals because of Roundup and some of its products. But one of the interesting things here, kind of returning to the discussion I was having about glyphosate, is that glyphosate itself is, though it's very effective in doing what it does in disrupting it, it's not completely soluble in water without being in a salt form. And that makes sense because water's polar, and when there's charges in there, it can dissolve that solute better. And so when these additives and a lot of other things are put into Roundup and some of these other herbicides, it can cause detrimental effects that have not been studied as well as glyphosate itself. So there are additives called ethoxylated formulants that are put into Roundup in order to increase its uptake into like leaves and plant cells. And in this study, it shows that in the presence of these additives and ethoxylated formulants, human cells quickly die and can have a couple of other really bad side effects. And a lot of other sources that this paper states cite studies that they've done saying that cancer and a host of other issues can be caused by glyphosate. Not to mention the fact that a lot of glyphosate is being detected in rivers. Since it is so commonly used, it accumulates and is one of the more prevalent organophosphates that are being found in polluted water these days.
So is glyphosate actually classified as a carcinogen by anybody?
Glyphosate is, I understand what you're saying, but glyphosate is classified by the World Health Organization as potentially carcinogenic, which kind of makes sense because they're looking out for people's health. But for example, the EPA in America is not registered as carcinogenic, and that's why it is still allowed to be used in America. And they are repeatedly investigating glyphosate, but until now they have not found any detrimental effects. And one thing that some of this work that I've been doing and work that other people are doing as well shows that instead of studying the glyphosate, maybe we should focus more on the additives that are also put in there.
So for kind of a solution to that, would you recommend they start looking more at additives?
They could. They already have explored a lot of different additives. Like I said, this herbicide's been around for a long time, and so they've really done a lot of studies on it.
So do you think that it's more of they've been complacent with the additives they found their cheap, effective method of doing it? Or do you think that, we could actually perform more science and find something that is actually less dangerous to humans?
I definitely think that we could look more into a safer way to apply it. But another thing that we should look into is how we use it. Like Roundup is a very effective tool, but does it need to be sprayed all over the place? How much do we really need to use it? And are we using it correctly? Is the dosage that we put on there proper? Do we put it on right before it rains? Like a lot of other things that we do as humans can cause harmful side effects in the environment.
Since you can use Roundup with no training.
Yeah, you can just go buy it and
Buy it at
Just go
Walmart
buy it
and put it on,
use
right?
it. So you might be using incorrectly if you're not following any labels.
It definitely has the directions on there, but who's to say that you're using it correctly?
Well,
And
reading's for suckers.
true.
But I mean, again, this is part of what we want to stimulate thought on is how are there other solutions for some of these pollution problems? Bailey was discussing how we have too high of regulations that maybe her papers are trying to raise more awareness towards what the actual limits should be, considering we get sick at a quarter of the value that's actually legally allowed in the EU right now. And
Huge problem.
something that we could look into with wastewaters. There are more expensive filtration and better filtration and separation options out there, but it's more of a cost-benefit analysis right now. Is the danger being presented by the chemicals we're allowing to flow downstream high enough to warrant these more expensive reverse osmosis filtration, for example? Is it too expensive to continuously run the large quantity of water that we need every single day through something like this? And this is...
It's also a huge problem everywhere, including in like Wyoming. We've even seen in like different problems with pollution, including like going back to the blooms, the algal blooms. We've seen some in Glendo Lake even. Like it is definitely spreading as a problem and needs a solution.
Yeah, I mean, just the other day, my mom was telling me about some of the fishermen, because she's a dental hygienist. She talks to a lot of people. One of the things she was discussing was just a lot of the people that go fishing. They've noticed a lot more algal blooms this summer than what they've seen in the past. And so, I think it is part of our responsibility to begin to look into some of these more effective solutions and more effective ways of separating things from one another. And I think that's kind of all the time we have for today, guys. I appreciate both of you for joining me.
You're welcome. It was
Yeah,
it was a
you're
pleasure.
welcome. Thank you
I really
for
enjoyed
having us.
discussing water, and hopefully we can find more solutions to the growing problem. Lots of rivers these days are being polluted and used poorly, and even the simplest things like where does the water that you flush down your toilet go? What are you putting in your toilet? Like even those simple problems or that
even
mindset can
how
change.
can I use pesticides and like herbicides more effectively where it's not getting into our groundwater?
And just a general idea of responsibility.
Yes,
And being able to
and
be
how
more
we can take
accountable for what we can do.
Yeah, and maybe glyphosate isn't the best thing to be using. Maybe we could find a better herbicide that doesn't.
Require so many additives.
Yeah, that is just as selective like glyphosate is. It only does one thing, which is good, but doesn't need to cause problems as well. Or we just use less of it. There's a lot of different things we can look into.
All right, guys. Well, thanks again for being on here. And I hope this has sparked some more inquisitive thought for all of you who are listening to this today. And as we continue to understand our knowledge and continue to grow and develop our understanding of what we are doing to water and why it is so effective, we also need to understand that some of these dangers and risks that are associated with it. However, I believe that these discoveries open up the door for those who are able to accept this challenge and be able to rise to this occasion that we can solve these problems. It won't be easy, but I believe that if we are not complacent, we can do a lot more for our most precious resource. So I'd like to close out with one more final thought. What can all of us do better to limit our own water pollution? Individually, we are strong, but together we can overcome mountains. Thank you for listening wherever you are, and I hope that you're staying happy, healthy, and hydrated. Thanks.